Hello, my name is Ahmad Afif bin Ahmad Nazmi, S50156. Today, I will explain to you about DNA barcoding in fisheries product. Let's get started. DNA barcoding is a method of species identification using a short section of DNA from a specific gene or genes. DNA barcoding technology also allows for species identification by taking advantage of a DNA sequence fragment that is shared by organisms that have significant interspecies difference. There are several steps must be followed in DNA barcoding. Problem: fish is a major source of protein for humans. There are many seafood products available in the market such as fish ball. There are some seafood products that mention the type of fish species to make the product and other does not mention, such as fish ball. So, what is the fish species that have been processed to make fish ball? The DNA barcoding will give the result on what type of fish species that have been used to make the fish ball. There are several steps in DNA barcoding. Step 1. Preparing the sample. Steps 2. Extracting the DNA process. Step 3. Polymerase chain reaction (PCR). Step 4. Gel electrophoresis using agarose gel. Step 5. Sequencing. Step 6. Result. Step 1. Preparing the sample. The fish ball will be the sample in this experiment. The small pieces of fish ball is taken and weighed it. Then, place the sample in empty Eppendorf tube. Step 2. Extracting the DNA process. The DNA extraction process frees the DNA from the cell and then separate it from cellular fluid and proteins and result in left with pure DNA. The three basic steps of DNA extraction are lysis, precipitation, and purification. Step 3. Polymerase chain reaction (PCR). PCR, or the polymerase chain reaction, is a chemical reaction that molecular biologists use to amplify piece of DNA. This reaction allows a single or a few copies of DNA to be replicated into millions or billions of copies. Step 4. Gel electrophoresis. Gel electrophoresis is a technique used to separate DNA fragments according to their size. DNA samples are loaded into wells indentation at one end of a gel, and an electric current is applied to pull them through the gel. DNA fragments are negatively charged, so they move toward the positive electrode. Step 5. Sequencing. DNA sequencing is a laboratory technique used to determine the exact sequence of bases A, C, G, and T in a DNA molecule. DNA sequence information is important to scientists investigating the functions of genes. This order or sequence of these building blocks of an organism's DNA is unique to each organism. So, knowing the order of the DNA of a sample allows scientists to identify its species. Finally, we can identify what fish species that were used to make fish ball that we eat. Thanks for watching.